Okay, so good evening. This is the last session for the course on basics of biology. As you already know, I am Ankita, and today what we will discuss is the week 12 discussion. So as you are already familiar with the video lectures as well, majorly this week was about more of a summarizing and conclusive discussion about everything that has been brought up in the past 11 weeks. All of that is summarized so that you get a holistic idea about any living organism as a whole. All of the different aspects that we have studied over the past 11 weeks, they come together and basic conclusions that you could draw from it as well. So because of the nature of this week's topic, first we'll go through the week 12 assignment, look at the questions that were given, and then we can try and catch up on as many of the topics that were previously discussed, just for a more of a revision sort of perspective for the past 11 weeks. We'll go through those lecture notes only, and then we can wrap up the course and you can proceed for your preparations for the examination. So as you already know, in week 12, the assignment obviously has been already done. So in week 12 assignment, what you were first asked, I'm sorry if this is a little hazy, there was some issue, but the first question was about the initial lectures where we had discussed about organisms and their diversity, their classification, etc. And there we had learned about this particular part. I'll read out the question first. So here we are asked that the body of the earthworm is divided into segments of serial repetition with few organs called as A, metamerism, B, isomerism, C, symmetry, or D, pseudometamerism. So as you can see, it's a little shifted, but as you can see, the correct answer for this is metamerism. Metamerism is the, you can say it is the phenotype or it is the characteristic feature majorly of arthropods because their body appears segmented. It is, of course, bilaterally symmetrical, which means that if you cut an insect body into half, you'll get two identical halves. So apart from that, it is also, they also have lateral appendages, which means that they have lateral, literally the terms mean, lateral means that it is on their sides. Appendages mean limbs, basically, like arms or wings or legs, whatever. So these, apart from these features, the next feature that is majorly of focus when we talk about arthropoda, we are talking about arthropods because your earthworm is mentioned in the question. So for them, the next characteristic feature is that they have metameres. They have a segmented body, which means that if you look at the body of an arthropod, you can divide it into sort of compartments, and these compartments mostly are exactly alike in terms of what they are made of. So same number of appendages, same feature, same shape and size more or less is present in them. So they are exactly, you can think of it as equivalent to how a cell, it forms a layer, like it forms a, say, a tissue layer, a squamous epithelium, which is made up of multiple cells which are lined one next to the other. So similar to that, analogous to that is the feature where in case of earthworms or in case of any arthropods in general, these individual segments, which are called as metameres, they are arranged one after the other in a serial repetitive manner. And what they constitute of is the entire organism. And this feature is known as metamerism, which answers your question. In question 2, we are being asked that the type of association seen between humans and the bacteria that live in our intestines is A, parasitism, B, commensalism, C, mutualism, or D, competition. So since you have learned a lot of different aspects of any living being about basics of biology, a very logical question that would come up is that since we understand that bacteria are not, under normal circumstances, not something that our body would allow because we have an immune system in place, right, which recognizes bacteria as pathogens. So it will recognize it as an attacker and will try to eliminate these species. So despite that, why are bacteria allowed to live? The reason for that is because, as you know, if they're living in the intestine, they're helping in the process of digestion. So here your knowledge about bacteria, how they are different from humans, which came from your diversity and classification lectures. Then comes your understanding of the immune system, which came much later, and your understanding of the digestive system. All of that merges, and from that you have to draw the conclusion and understanding that if bacteria are living in your gut, if your immune system is allowing them to live in the gut, it must be providing some benefit, right? And now we know, studying digestive system, is that these are very necessary bacteria. They are called our um, gut microbiota, which means these microbiota simply means microorganisms. So these gut microbiota are absolutely essential for us because they are necessary for the digestion process to be completed. Because we don't have all of the enzymes, all of the we don't have everything that is required to digest all that we consume, and a large part of that is taken over by these gut microbiota, and therefore having healthy gut microbiome is extremely important. So if they are providing some benefit to us, and because our bodies are allowing them to live within us, it is a sort of equation or association where both the beings, both the parties, for lack of a better word, are benefiting from each other. Hence the relationship is symbiotic, and in particular it is a mutualism based interaction where both are like, if you have gone through the matrix of different types of symbiotic associations, where if you consider party A and you consider party B, then the equation can either be beneficial and then it is denoted as a plus, or it can be harmful, then it is denoted as a minus. So since both parties are benefiting each other, it is a plus plus sort of equation, and therefore it is mutualism. If it was parasitism, then what sort of an equation it would be? It will be different. It will be an equation where party B is taking over party A, as which is the host, where party B is getting benefited. Obviously, it is deriving some nutrients, shelter, any sort of benefit it can be. But party B is suffering as a result of it. Its health is deteriorating, and they are, in many other ways, they're getting harmed, right? So that sort of an equation is plus minus. So you can look up each of these different types of other interaction as well. Commensalism and competition also are different here. So that completes the domain of question 2. In question 3, we were asked that the absence of nucleolus is seen in A, fungi, B, archaea, C, protista, and D, animalia. So the nucleolus, if you remember your initial lecture notes about cells, right, the basics of cells, then you would remember that within the nucleus, you have some sort of compartmentalization. It's not very distinctive, but it is there. And within the nucleus, there is a smaller portion which is annotated as the nucleolus. The remaining part is the nucleoplasm. And this depth of classification is seen in relatively more, I wouldn't say advanced, but somewhat more complex organisms compared to archaea, which, as you already are familiar, they're very, very primitive organisms. They do not have a defined nucleus at all. And therefore, there's no question of a nucleolus really being in place. So fungi, protista, and animalia do possess nucleolus, whereas archaea do not. And that is basically your question number 3. Then in question 4, we were asked that the force with which coacervates are held in the formation of colloidal aggregates is. So here, I don't really have much to explain about it. If you remember, it was we had an entire discussion about the origin of life, how organisms might have evolved from where they would have started, where they ended, etc. So it was a direct question over there. So the type of force which would actually operate in the formation of colloidal aggregates is hydrophobic in nature. So that would be the hydrophobic force would be the answer to your question here. Then in question 5, we are being asked that who considered evolution as the hierarchical linking of series of forms in Scala Naturae. So this, if you remember, I think we discussed in the very first interaction session as well, is that before your exams, you should definitely go through some of the most eminent contributions made by some scientists from the field of biology. There would be hundreds and thousands of such examples, but these are some of them you'll find in your lecture notes. Some you can just look up on Google. You can look up recent Nobel laureates. You can look up scientists with with contributions that changed the face of biology. So you can look up, it'll be good for your own learning. And of course, it will also be, could be also very beneficial for your future as well as for this immediate examination as well. So here we are talking about Scala Naturae, and that was written by, or that was a thought brought about by Aristotle. In question 6, we were asked that euglena, they reproduce by the process of A, fragmentation, B, binary fission, C, budding, or D, regeneration. So if you remember the example, so you'll have to, as you can see, a lot of questions actually are related to the very initial topics, particularly from diversity and classification of living organisms. So make sure you go through them really well. You try to brush up on the initial topics, because students have a tendency to kind of neglect them because they were in the earlier classes and you feel that you've learned a lot more advanced concepts, so you don't go over these. But that's not a good idea. So you try and go over these topics again. And in these classes, we have discussed the different types of, based on the increasing complexity, how the process of reproduction of cell division, etc., all of that changes as well. So there you'll see that euglena are capable of reproduction by the process of binary fission. So that would answer your question. Then in question 7, we're being asked that the membrane that strengthens molecules in prokaryotes is. So I think this is a relatively less, a less familiar question for everybody. So here the answer is hopenoids. Cholesterol, sterols, and isoprenes are relatively more known to you. In question 8, we were asked that the phase at which the spindle pole position checkpoint that is present during the cell cycle is A, S phase, B, G1, C, G2, or D, the M phase. So if you remember the different phases of the cell cycle, then so how do you tackle a question like this, right? If you remember each of these stages and some characteristic feature from each of these stages as well, then you'll remember that if you look at what the question is asking is when is the checkpoint that makes sure that your spindles are correctly aligned. When is that actually coming in? So in the entire cell cycle, the only phase during which the spindle actually assembles. What is the spindle? These are relatively more advanced topics, but basically spindle fibers, they attach, if you remember the haploid chromosomes during metaphase, they align right along the metaphase plate. And from each of the two centromeres, centrosomes, what you get are extensions, little pipe-like extensions, which are microtubules, which is a part of the cell cytoskeleton. They attach to the chromosomes and they allow equal separation of the chromosomes to each of the two daughter cells. That is its job. So whatever is the genetic material of the initial mother cell, say it is X, then that it gets replicated during the process of DNA replication to become 2X or 2N. And during the process of cell division, it becomes N and N once again. So this is what the spindle position the spindle pole checkpoint is actually doing. What it is ensuring is that each and every chromosome that is present within the cell, that is definitely attached to the centromere. So if you have, for example, we have 23 pairs of chromosomes, right? So the cell always makes sure that each and every chromosome, like all 23 pairs of chromosomes, they are attached via these microtubules that I just mentioned. You can think of them as threads or as some sort of a pulley chain or some sort of mechanism that is operational there. So it makes sure that each and every chromosome gets attached so that you can ensure that each of them will get separated equally. If under some cases, if some situations, there is not, or there is some, this checkpoint has failed, or there is some sort of a reason for which it has not managed to attach all 23 chromosomes, then what happens is that in one of the daughter cells, both copies or both chromatids would go, or both sister chromosomes would go, whereas in the other copy, it would lack one copy of the chromosome, right? So you would have, say, if we are taking the examples of human only, so 23 pairs of chromosomes, of which one particular, or you think about autosomes in particular, 22 autosomes are present, right? So among them, one pair has not correctly attached. Then what would happen is that in each daughter cell, they would get 20, like one daughter pair would get 22 plus two, like an entire chromosome. On the other hand, this would lack, the other daughter cell would lack one chromosome. So that would be a deficit, and these conditions are known as non-disjunction, which means there is an inequality in the distribution of chromosomes, and that leads to really, really severe disorders. I think some people might have actually come across is that when it occurs in case of sex chromosomes, then it leads to some really severe diseases. And also for autosomes as well. Like there's Turner syndrome, which is for sex chromosomes, Down syndrome, trisomy of 21, right? So these are cases where any sort of disjunction problems, when they occur, they lead to really severe diseases. So this checkpoint is extremely crucial, and it makes sure that if it is not attached, it will not allow the cell to divide, and it will keep a check on it. So all of this will most definitely occur during the M phase. So we deviated from the question a little bit, but I was trying to give you some context as to why it is so important. And the phase that actually is playing the major part, or the phase in which this is playing a major part, is the M phase. In question 9, we were asked that the first ever genetic recombination mechanism in bacteria was done by, so Griffith's experiment, if you remember. I'm not getting into details of this. We'll actually go through the topics when we brush up over it once again. We'll go through it. But yeah, Frederick Griffith was the first person who tried recombination, or understood the recombination bacteria. Then in macromolecules, where we took up carbohydrates, proteins, all of these one by one. So in case of the neutral lipids that are most, something is wrong with it. Sure. But yeah, so the most abundant neutral lipid was it's triacylglycerol, diacylglycerol, monoacylglycerol, or cerebroside. So of course they are triacylglycerols. They are the most common. They make up a large part of your adipocytes or fat cells, and that was the direct answer to this question as well. In 11, we were asked about reducing agents. So the reducing agent that reduces disulfide bridges in a protein into thiol groups is A, beta mercaptoethanol, the B, guanidine reagent, C, urea, or D, alkaline. So disulfide bridge reduction is majorly done by beta mercaptoethanol. Another reagent that is also very commonly used is dithiothrietol, or DDT. These are the two most commonly used reducing agents for. So you imagine this is the 3D conformation of a protein. If you just give it heat, or you give it SDS when you are trying to run an SDS page, then it will get somewhat linearized. But if at any place there are any disulfide linkages, those will not open up. So the protein will not be completely linear, and therefore whatever results you're getting is not entirely reliable. Therefore, you need to have a reducing agent which will make sure that no cysteine disulfide linkages, which leads to the formation of cysteine, and that significantly changes the protein conformation. No such disulfide interactions should be present in the protein when you're running assays like this. So for that, you majorly used is beta mercaptoethanol to perform such what should I say, such normalization before you actually perform an experiment. In question 12, we are being asked that the only aromatic amino acid with an ionizable side chain is A, tryptophan, B, tyrosine, C, phenylalanine, or D, cysteine. So I don't really have much to say about this, but yeah, if you remember this, okay, so for this, even if you don't completely remember all the amino acid structures, it's okay. But there are a few relatively less usual amino acids like tryptophan, tyrosine, phenylalanine, histidine, and proline. So these amino acid structures, if you are able to recall them or at least practice them, because they are a little unusual from the usual pattern, general pattern. So it's better to keep your at least keep it in mind, even if you don't really practice it much. Tyrosine is relatively easy, just has a benzene ring with an OH group. So that OH group is ionizable. It can donate an H plus, it can accept an H plus, and therefore that's an ionizable side chain. Sorry, just a second. So in question 13, we are being asked that the technique that is used in identification and quantification of preservatives and sweetening agents in food is. So when you are trying to quantify something, so okay, even if you don't completely know this answer, because this not necessarily taught in those, even if you, or if it was, something you don't necessarily know, then under such circumstances, what you do is you go through each of the techniques. You have X-ray, you have ST stage, you have ELISA, and you have thin layer chromatography or TLC. So what is being asked is, if you are trying to understand and you're trying to quantify some agent, some chemical which is present in some food material, right? So X-ray cannot quantify anything in food material. X-ray is used for solving crystal structures. It's a very high, it's an ionic radiation which is passing through your, and based on that you are understanding the diffraction to understand the protein structure. So it cannot identify or quantify anything in food. SDS page, again, this is why I'm stressing that you have to know the technique even if you don't necessarily know the exact answer to the question. SDS page is used for resolving proteins, right? You have a mixture. If you take an entire cell lysate and you have hundreds and thousands of proteins in it, from there you allow it to separate, you allow it to resolve, and from there you identify some protein which is of your interest, right? So that cannot be done for food materials. It cannot address the question this particular situation is asking you. Neither can ELISA. ELISA has a completely different objective. It is an immunotechnique. It is used for specifically identifying one epitope or one reagent from an entire mix. So thin layer chromatography would automatically become the default answer you opt for. So this is how you tackle questions if you're not entirely sure about what the answer could be. Don't just randomly guess. It can lead to, it can cause you more harm than good. But if you can logically arrive at an answer to a question, definitely do it. Then in question 14, we're being asked that the histone acetylation, which is catalyzed by histone acetyl transferases or HATs, reduce the what you need to, this is also a very basic question. What you need to understand is that if the enzyme is a histone acetyl transferase, what is it doing? It is transferring acetyl group to histones. That in itself, it gives you the answer directly. But if you think about it a little more logically, the remaining part of the question is just confuse people. If anybody have lysine arginine residues, etc., so they are very, very positively charged, whereas the DNA that is bound around them, that carries a negative charge from its phosphodiester backbone. So the two of them have they're attracted to each other, which maintains the heterochromatinized packaging that you are obviously quite familiar with. So when a histone acetyl transferase is coming in and it is transferring an acetyl group, which if you understand, it carries a minus charge, right? So minus charge. So when it is imparting a minus charge onto the histone, what is it doing? It is reducing the positive charge, and therefore the binding with DNA will get reduced because negative charge on histone will repel the negative charge on DNA, and the two will no longer have a tight binding. And those regions, they sort of open up and they become transcriptionally active. So this is one of the major mechanisms which are epigenetically controlling the expression of certain genes. So that is how it works. In question 15, we were asked that the N-linked glycosylation in eukaryotes is inhibited by A, telithromycin, B, clindamycin, C, tunicamycin, or D, chloramphenicol. So if you went through your lecture notes carefully, you would get a table of the different types of drugs and what they do. And apart from that, also for your own knowledge and for better understanding, you will find a lot of such tables and composite materials that show how different antibiotics they work. So not just antibiotics, you can go through antibiotics, you can go through some plant derived products and how they work. So you will be like it would be fascinating to you to see how something derived from a plant is able to do such insanely difficult tasks. And you'll be also be able to understand how we as humans have evolved to utilize different organisms, their products, their byproducts, etc., to fulfill some completely unrelated purpose. And that in itself is an accomplishment of human beings as a whole. So but the point of the matter that I got deviated from was that go through some of these antibiotics, go through particularly their site of action. Because, for example, if you go through antibiotics, target transcription or trans eukaryotic organism. Even under translation, site of action can be different. It can bind at the channel. It can do something else. Puromycin works in a completely different way. So the different ways of action are also different, and it would really be helpful. There are also some questions that are extremely relevant, so just try to brush up on these a little bit. Then we were asked that the frequently activated death protease that catalyzes specific cleavage of many key cellular proteins is A, thermolysin, B, ADAM12, C, caspase-3, or D, elastics. So there is an entire set of proteins that actually work. It's not just one. Even though it's written as a death protease, it has a major role to play in cell death, but that's not its only job. But the entire caspase cascade, the reason it is called a cascade is because there are a chain of enzymes which act one after the other. And once a cell has encountered a situation where it is understanding that its best option is to die, what it will do is it will trigger apoptosis. So apoptosis is the process of programmed cell death. That the cell, it opts for mechanisms which execute its death in a very controlled manner without like lysing and releasing all its contents into the exterior. It does everything in a more controlled fashion so that it can utilize most of its resources, utilize them efficiently while conserving as much energy as possible. And this entire process, one of the ways in which this entire process is initiated is via this caspase cascade. So multiple cascades, you can just look up the entire cascade just for your own reference and see how even the mitochondria is involved in it. And surprisingly so, which provides energy to your cell, but it also helps a cell to die when that is the requirement of the cell. So you can go through things like this for your own understanding. It'll be helpful for you. In question 17, we were asked that the glycosidic bonds in starch get hydrolyzed by A, trypsin, B, alpha amylase, C, amylase, C, mucin, or D, pepsin. So if you remember the salivary amylase, you might have, like if you remember your digestion classes, you would recall that, carbohydrate digestion, it begins in the mouth. So the reason for that is because you have salivary amylase, which is in your saliva, and therefore it helps break down the sugars right from the very beginning. And that is what is happening here as well, that in starch, the glycosidic bonds, they get broken down, and alpha amylase is the reason for it. In question 18, we were asked that the like projections that are please know to do much, because this is something you've been learning for a very long time, for many years. So the answer for this is villi. They increase effectively the surface area for absorption more. So if you have a flat surface, the surface area which is present here, versus if you have present or this is operational within the intestine. So villi are exactly the structures that help in doing the same. Then in question 19, we were asked that the casein protein in dairy products are digested in stomach by A, chymotrypsin, B, trypsin, C, elastase, or C, carboxypeptidase. So casein digestion, it begins by trypsinogen, trypsin, and chymotrypsin, all of these different enzymes, they work, and they help in the entire digestion process. Renin and casein get digested differently. So that is the cascade that if you remember your digestion classes, it was mentioned there. We'll probably go over that as well. In question 20, we were asked that the deficiency of factor 9 during blood clotting results in A, von Willebrand disease, B, hemophilia C, C, hemophilia B, or D, hereditary angioedema type 3. So this was also a tabular sort of chart which was provided where the defect in each of the different factors was 9 deficiency leads to hemophilia B. In question 21, we are asked that in relatively recent classes, so you might actually remember this as well. So the active part of the or the active brain, it shows a different wave pattern. So in that case, you would detect beta waves, and lack of beta waves is usually it's not necessarily a worrisome situation, but an actively thinking person would have very whether they're thinking actively, whether they're asleep, all of this can be studied at different brain waves. So that was question 21. Okay, I completed here. So in question 22, we were asked that the resting potential in neurons is attained by taking in. So if you remember the entire process of an impulse generation and all of that, you would remember that the inside of the cell is always negative, and this inside negative status is maintained by exchange of sodium and potassium. So the balance that is maintained is such that the cell always sends 3 sodium ions out and it takes 2 potassium ions in. So this actually maintains the basic membrane potential, basic neuronal membrane potential, or electrochemical gradient is maintained, and that is because it keeps on it's sending Na plus out and taking K plus in. So that would answer your question, that K plus being taken in is the reason why you are or the cell, particularly neurons, are able to maintain the resting potential. Just a second. Sorry for the disruption. Yeah, coming to question 23, we were asked that the nerves of face and saliva secretion, they originate from, if you remember the different parts of the brain, the different functions that we had already discussed. So this actually comes from the medulla oblongata. These are actually quite detailed questions, so I don't really have much suggestion in this regard. What you can do is when you tackle such large variety of questions, Some areas you try to strengthen to your best possible capacity. So always get a scattered pattern of questions. And what you can do is you can choose whichever questions you're most confident in, answer them first, then move on to questions you're a little uncertain about. And if you're absolutely not sure about any questions, try and avoid those. Of course, in situations where no negative marking is there, that is fine. But if you are in the future in any examination where negative markings come in, you should learn to avoid questions like that. So this is a question where you need to simply remember, and sometimes your recollection can be a little dicey. So that is how you would have to deal with situations like this. So yeah, the answer to this question is the medulla. In question 24, we were asked that the mineralocorticoids, they increase blood volume by increasing sodium and water reabsorption and potassium excretion from, answer here is C, kidneys. And obviously, because the reabsorption of water, of sodium, and whatever is basically the control over how much concentrated an individual's urine would be or would not be, it is maintained at the kidney. And a lot of hormones, as you've already understood during the homeostasis discussion as well, they actually have the role in controlling that, and that occurs at the level of kidneys. And finally, in question 25, we were asked that the receptor that causes a change in the stimulus, if you remember that entire, that is also a cascade, that cascade of different sub-steps that operate in executing a particular action. Then the level at which signal is detected, which is a change is sensed, is done by the sensor. So that would answer your final question. So I think with this, we are completing the assignment part of it. In case of you have any questions, please ask, or if you have, if you want to write it down, go ahead and do that. And in the meantime, I will open up these initial discussions, these week-wise discussions, sorry, and we can just browse through as many as we can cover, because obviously we have discussed this previously. I'll just go over these so that you get a revision sort of a flavor here. So bringing you back almost 3 months, the initial lectures, they talked about science, how it can be divided, the different domains of it, how natural sciences are divided, and in particular, how we are currently talking about life sciences in particular. And then here, like I already said, so the different contributions made by some of the most eminent biologists, scientists, could be Nobel laureates, could be some very fundamental discoveries that they did, could be some life-changing techniques that, for example, PCR, Caramelis is credited with bringing about this revolutionary technique. And that's an immense contribution to not just the field of biology, to health sciences as well. Like, if you understand, if you were in the, you were aware about what was going on during the entire pandemic, you understand that all of the tests were carried out by the process of, or by the technique of RT-PCR. So these are revolutionary techniques which are extremely, they have a huge impact on mankind. So remembering these scientists and understanding how huge their contribution truly is will only do you good. So that is what we see here. So there was Aristotle, Galen, Malpighi, Harvey, Leeuwenhoek, Darwin, and all of their different contributions. Then here we see the different divisions of biology and what are the different aspects. If you really try and compare them, then the living objects and the ability of a living object, which starts from self-growth, its ability to produce energy, to replicate, produce more of its own kind, and to move. Most living organisms are motile, of course, with plants being the exception to this. And then we move towards the idea of diversity. We understood why diversity is required and why it is necessary to actually understand it. Why are we really studying about it? So there, if you remember, I had given the example of how, for again coming back to PCR, for performing PCR, the enzyme that was first used was isolated from an archaebacteria, which was Thomas aquaticus. So you never know where you would find inspiration or where you would find resources. So that is why such intense understanding is quite helpful. Then we understood how organisms can differ in terms of their shape, size, where they're distributed, what is the biological activity, and so on and so forth. And in the upcoming weeks, we took up all of these different discussions, and we'll go through some of them now as well. There's some issue here. It's fine. Yeah, it's fine. So yeah, then again, coming to why you should classify, because you need to understand and study them. And the more you simplify the entire living world, the more it gives you an idea about where you are in the boat and how you can really derive the best you can from the resources that you have. And also to understand your own origin, your evolution. When I say own, I mean of mankind as a whole, but yeah, for those purposes as well. Then additionally, for health benefits as so also, like understanding how vaccines can be developed, how you can get drugs, you can get drugs from plants, you can get inhibitors, you can get some revolutionary product from anywhere. So that is the idea behind doing classification. And then one of the most um, widely accepted as well as popular ways of classification, and these are the different bases on which he did so: cellular structure, body organization, the mode of nutrition, and of course how an organism reproduces. So this was fairly accurate. There has been development on this, and in more advanced studies on the topic, more and more types of classification. There are 7 kingdom classification, there is sub classification of each kingdom, and lots of different ways in which you can classify organisms, but this is the most easy to understand and the most I would say comprehensive way in which living world has been classified. Then in the 5 kingdom system, we have Monera, Protista, fungi, plantae, and animalia, and these are the different groups that are included under it. And here you see a comparison between them. Again, wherever you see a table in any of the lecture notes, make sure you go through it. There is a reason why it has been provided to you in that way, for your understanding, for you to just take a glance at something and be able to brush up 20 different things about that topic. So that would be a very, again, a very holistic, very comprehensive way of studying such topics. So here you will go through the cell type, whether they have a cell wall, what is the organization, do they have a nuclear membrane, what is their mode of nutrition, so on and so forth. And of course, examples for each. Then we went through each of the kingdoms. Monera, I'm not going into details. You've done all of this. We are brushing up. So we understood Monera. We went through chrysophites, their subdivisions. Then we came to kingdom Plantae, how they're suddenly completely different. They are multicellular in nature, and they have a cell wall. Their mode of dividing, the mode of nutrition is also completely different. In contrast to that, the kingdom Animalia is completely different. They have different organ systems. They are a lot more complex. They're motile, so on and so forth. And we found some exceptions to this as well, like viruses, viroids, and lichens. Then how organisms can be actually, the nomenclature can actually be given to them. So that was basically what was covered here. Sorry, this is taking some time. Yeah, then we went through each of these different criteria. Actually, I'll not get into details about this. I'll just go through this part. We talked about the different levels of organization, whether it is an organ level, organ system level, tissue level, what is the situation. That we talked about the different types of symmetry, whether it is radial, whether it is bilateral, or whether it is asymmetric. All of these different types of organisms we understood, and we understood that with more and more complexity. You will move towards bilateral symmetry, whereas in lower organisms, you will either find asymmetry under all circumstances. Then we understood how organisms during the process of their development, how many layers they form. Depending on that, also you can classify organisms as either diploblastic or triploblastic, whether they have an ecto, endo, and mesoderm, or they just have an ectoderm and an endoderm. So that's also another way of classification. Then in fourth way of classifying was segmentation, which is something we discussed just now as well, that metamerism is something you see segmented body. And finally, the a very, very large chunk of the entire living world, that I wouldn't say a large chunk, but yeah, fairly big chunk that goes into the classification of chordates has the name that arises because of the presence of a notochord, and you can see the clear vast difference between chordates and non-chordates as well. So that becomes a very essential way of classifying. So that was in depth, sorry, covered in this lecture for week 1. And okay, this is not opening. Yeah. Then, yeah, again, another tabular format of compiling all the information that was given. So you'll find the level of organization, the symmetries, the body cavity, whether they have one, and the different phyla which are included among them. And here you'll find all of the differences between chordates and non-chordates, and even under that, the subphyla that are present, how they are classified, examples of each, whether it is bonyfish, whether they're different features, then how vertebrates are different from them, all of that was covered in the initial lectures as well. Then we came to, yeah. Then we came to the plant kingdom, how flowering and non-flowering plants can differ, what are the different gradations or stages among them, and how they also evolve from less complex to more complicated organisms. So that was pretty much, I think, covered under week 1. Then in in week 2, we move towards the basics of cells, I think. Yeah. We moved towards evolution. We learned about evolution of the earth. We understood first how the earth is. We understood how life has originated on earth. What are the different theories, some of which rely on some special power, some of which rely entirely on appearing out of nowhere. Some of them rely on religious beliefs, etc. But what we majorly look at is the modern theory and the theory of spontaneous generation, and all of these different other hypotheses have been disproved as over the course of time. So each of these theories was looked at in quite some detail. We looked at some experiments. This is a particularly important experiment about Reddy's experiment for understanding the theory of spontaneous generation and how it was technically not feasible. He eventually ended up disproving it, and it indicated what is proposed by Schleiden and Schwann as well, two very eminent scientists again, who said that life cannot appear out of nowhere and cells always arise from pre-existing cells. So that is how they really operate. And there was also Spallanzani's experiment for this as well, Pasteur's experiment. These are all very, very crucial experiments. Please make sure you go through them. And these are the other theories that were put forward. There was some positives and negatives that were attributed to each of them. And of course, based on that, the modern theory has a lot more scientific backing to it, and it is more accepted, you could say. So these were other experimental evidences. Particularly, this one is really, really important. It is the Miller and Urey experiment. Please make sure you remember everything that is given in this experiment. It's not just for an exam purpose, but this is a very crucial experiment in the entire, I would say, one of the most important experiments in understanding evolution of living beings. So the concept, what are the different components that were understood, what was the conclusion that was drawn from the experiment, all of this you should try and understand to your level best. Then these were the other gradual events that were understood, how inorganic molecules were formed. From there, you got organic molecules. From there, how you suddenly got simple organic compounds, more complex organic compounds, sugars were formed. Then came coacervate formation. In fact, this was a question that was also taken up. And we were asked that how are coacervates going towards aggregates, and what is the type of force or interaction that is present there. So it was hydrophobic in nature. And then it eventually leads to the formation of eubionts and protocells. And from there, we completed the idea of how from nothing, from, I wouldn't say nothing, but from chemicals that were just present and the surrounding conditions of the earth, which were prevailing at that point of time. A lot of background noise. Yeah. So how from those chemical or those inorganic molecules, finally, the first cells were actually formed. Yeah, so that's basically what was looked at. Did I close it? Okay. Yeah, sorry. So then we came to the mechanism of evolution, the different scientific evidences that are present. Then the different ways in which it was understood. We had Lamarckism. We looked at some supports and some evidences that very loud. Sorry. So some evidences that supported it, some evidences which went against the entire idea of Lamarckism about acquiring characters based on the situation that an organism was faced with, and how over a period of time such acquired characteristics they would fade away, or whether they would get passed across generations. There were some examples that support, like for example, in case of giraffes, he said that their neck was so long to adjust to the availability of food, which was at a higher level. Whereas evidences against it were, for example, for Chinese women, how their feet are always were always kept in very uncomfortable, very tiny shoes to make sure that their feet were small. But obviously, that did not get passed on to the next generation. So there were evidences that were provided in concept of this. Then came Weissman's theory and neo-Lamarckism that also came in, and then came Hugo de Vries's theory, or the mutation theory, that was heavily speculated upon, understood, and is quite widely accepted as well. Yeah, the mutation theory, then how Charles Darwin and Wallace, they went on their voyage and they found a lot of data as well. How he wrote Origin of Species, etc. Then the theory of natural selection was brought forth. There are some concepts on which natural selection is based, and here you can see a large number of them, not going through them. Then came some evidences that actually supported it, like artificial selection, mimicry, protective coloration, etc. But again, there were some evidences which were going against it. So that is, when a hypothesis is brought forward, you will not always find everything which supports it. You will always find some negatives, some positives. If you can defend those negatives, your hypothesis could be acceptable, and it has to be statistically significant as well. So a lot of technicalities come in, after which finally the conclusion can be made. Then came Hugo de Vries's mutation theory that we were talking about, his experiment that he performed with evening primrose as well. And from that, the conclusions that were drawn in. So make sure you go through this. Again, a very important chunk of this entire lecture as well. So that was for week 2. In week 3, yeah, in week 3, we took up cells as a topic. Again, I've discussed quite a bit about it. Here, go through this table. It is quite simple. It would take much memorization or just blindly remembering things. But instead, yeah, it is logical. You will find very clear differences between them in terms of size, genetic material, replication, um, etc. And just to go through each of these basic differences between the two. Then here you see the characteristic features, like flagella, like you see here, the surface layer, which is present in bacteria, the cell wall presence of the peptidoglycan, the two types of cell walls, because of which they can be gram positive, gram negative. What is the basis of the difference? How would you identify the two? What are some examples of different types of pathogens which are under either category? All of that could be understood. The compositional difference also has been provided here. Gram staining has been explained here. Then we came to the cytosol, the chromosome, extra chromosomal DNA present in case of prokaryotes as well, different types of plasmids, the different conformations of plasmid, how they migrate at a different pace, or you can say that they show differential electrophoretic mobility when they run through an agarose gel. So all of these are like a huge number of differences. Yeah, then the presence of different regions within the plasmid, how they have different functions to perform, like an origin of replication, selectable markers like antibiotic resistance genes, etc. All of that was provided. Then the protocol for plasmid isolation by mini, midi, or maxi can be performed. Each of the different reagents three different solutions, solution 1, 2, and 3, they're different components. What is their job? Why they're used in that sequence? Why in some places you vigorously shake a tube when you're carrying out plasmid isolation, whereas in some case you just invert mix and place it. Some steps you vertex, some place you do not. All of that has significance, so that what you finally get is pure isolated plasmid and is not sure genomic DNA. So all of that can be ensured. So this is the plasmid isolation protocol. And yeah, what you see here is an example of an agarose gel. And what you see at the bottom is RNA, because here you could not, you just isolate a nucleic acid. You cannot isolate or you cannot exclude RNA at this step yet, unless you use some enzyme to digest it. So that was the first portion. And then we came to the eukaryotic cells and some distinguishing features, but mainly between plants and animals. Some of them are physical differences, some of them are functional differences. They have different requisites, etc. Then the function of the cytosol, the nucleus, which is the main genetic content holder within the cell. Then the mitochondria, its different functions in not just in as a powerhouse, but also, like if you remember our recent discussion as well, just today in one of the assignment questions was about apoptosis and how the caspase cascade actually has the major role to play in it. But what you mainly associate, the main function that you associate with the mitochondria is still energy production. So each of the different protein contributors in this entire process, from complex 1 to complex 5 and ATP synthase, etc. All of them have different roles to play. So these are the different type of features which operate here. Then you have caspases and some detail about caspases as well. The idea given by Peter Mitchell of chemiosmotic synthesis of DNA, that was also provided. So that was covered in the second portion of this week 3. Yeah, then we talked about the chloroplast, the process of photosynthesis, the entire process of vesicular trafficking, which is like a traffic police which stands at the junction after a protein has been synthesized, and based on whatever is its address, it directs proteins to its correct destined destination. Because if there is any deviation or any incorrect sorting, it leads to very, very severe consequences. So that is certainly, at least it is attempted to be avoided. Then comes the ER, endoplasmic reticulum, its different functions like steroid hormone synthesis, detoxification, calcium sequestration, etc. Golgi bodies, their function, lysosomes, the so-called suicidal bags of the cell, but they have other really important functions as well. They degrade proteins that digest anything which is not necessary. Then the plasma membrane, its lipid bilayer nature, its composition, how it is not made up of only phospholipids, but also proteins, cholesterol, etc. Then, yeah, then sure you see pretty much the same. I'll move on to the next. Yeah, then cell nutrition and growth, how from polymers they are broken down into monomers, and the monomers are broken down into atoms, which can be used for synthesis of new products again. The composition of mammalian cell culture media, the process of cell cycle. We discussed actually a large part of this today. It's different phases, interphase G1, G2, S, and G0 phase. And apart from that, the most crucial M phase. So yeah, if you remember this particular step, which is during anaphase of mitosis or meiosis. So there you can actually see the microtubules extending forward and pulling those chromosome sister chromatids back. And then prophase, metaphase, anaphase, telophase, and cytokinesis were studied. And then finally, here the role of the cell cycle was understood in terms of cell growth, cell development, cell replacement, for regeneration, and for asexual reproduction as well. And what happens when any of these go wrong is that there is uncontrolled cell proliferation, which is obviously extremely harmful for cells, and it can be it can lead to cancer under certain circumstances. It can form tumors and etc. It leads to a lot of really major genetic effects. Yeah, that was week 3. I'll open week 4 and I'll just take a 2-minute break after that and we'll come back to it. Yeah, just give me a second, please. Yeah. Okay. So in this week, what we looked at was biological molecules or macromolecules, which were started with nucleic acids, lipids, carbohydrates, and proteins. So these are the 4 major um, groups, you can say, of macromolecules, which execute all of the different functions that you require for a cell to be living and be functionally active as well. So, oops. So that is what you see here. First, we started off with nucleic acids, all of the different experiments that are mentioned here for understanding that DNA is the genetic material of the cell. Please go through them. Make sure you conceptually understand them very clearly. They're important, which is Griffith's experiment, like I already said during the assignment as well. Then what is a nucleic acid made up of? It's different components, the sugar, phosphate, and the base. That is each of these components. How are they joined to each other? What are the chemical bonds that are present between each of them? Make sure they're very clear in your head. These are very simple things, but they get jumbled up when you are studying a lot of things and trying to sort out the correct answer for them. Then the structure of the nucleic acid. Yeah, that's pretty much it. What is the structure of DNA? How each of these bases are bound, they're binding to okay, yeah, they are interacting or they're forming a bond with each other. Let's say they're bound to each other. Why is there a base pairing which is occurring? What is the rule for it? Chargaff's rules which come in. So all of that, make sure you go through them. The control of cells. I think this was a incorrect slide, but yeah. The concept of self complementarity. well, not self-complementarity, complementarity, which exists in the two strands of DNA, which run anti-parallel to each other. And that is between a purine and a pyrimidine, which ensures that the structure of DNA is always maintained. And this is Chargaff's rule, like I was just talking about, the concept of DNA denaturation and stability, how you will get a value, the melting temperature at which exactly half of the amount of nucleic acid that you initially had, a double-stranded nucleic acid that you initially had, the temperature at which you'll get exactly half of that is the TM. Then the process of genomic DNA isolation, you please make sure that you understand the differences that exist between plasmid isolation and genomic DNA isolation. They're a little bit different. So the concept is at the end the same. You're trying to isolate one material from everything that is inside a cell. But yeah, the reagents that go into it, the steps that are operated, what you eliminate versus what you try to enrich, all of these are different. So that you need to study in a little bit of depth. Then DNA sequencing, the dideoxy or Sanger method of sequencing DNA, which is, uh, while it might be old, it's used till date. Modifications have come up in it, of course, but till date, it is used for sequencing short fragments of DNA, for sequencing regions that are important to you. But when you look at a more high throughput approach, you go for something like next generation sequencing, you go for whole exome sequencing, you go for whole RNA sequencing, or when you want more specific, you go for single cell RNA sequencing. So lots of techniques have developed now when large systems have come up that are able to sequence many samples at one go and efficiently do so. And these are called high throughput assays. But yeah, these initial relatively seemingly primitive methods are still very useful. They are performed in many laboratories by themselves. They're sent out for sequencing as well, and they're very important. So make sure you understand the concept, how a dideoxynucleotide, when it is incorporated, it stops the process of elongation, and therefore you can sequence that particular region. So that is the concept that goes behind it. And this is how you read through the gel, and make sure you do not get confused here as well, because you could just be given a gel like this and you just have to find the sequence of the DNA. So a lot of time people get confused between the 5' and 3' ends, and they end up writing the completely opposite sequence compared to what was intended. So please try to make sure that doesn't happen. And the Maxim Gilbert method is also the other approach towards sequencing nucleotide fragments as well. Here, a combinatorial approach is taken up. It is used, but I would say relatively less used. Sanger sequencing is the more popular way of going about it as well. And here you have the RNA isolation protocol. Remember the differences in reagents, the concept that is operational here, how trizol is required for the separation. So yeah, you can just brush up over these topics and you'd be good to go. So that was about the nucleic acid part of this week's lectures. Then came carbohydrates, how they have been used, how they are different, what is their role, what are the different formats, monosaccharides, disaccharides, polysaccharides, etc. How their nomenclature is carried out. Then the different properties like hemiacetal. From glucose, you will get the formation of hemiacetal in two different type of projections. You can depict it in two different ways. Then you have the hemiketal formation, which is you see for fructose. So these subtle differences you should also keep in mind. Then the two types of orientations also, whether they move towards the left or whether they move towards the right. Based on that, you get levorotatory or texturorotatory, indicated by L or D respectively, or plus, minus and plus sign respectively. Then the idea of enantiomers, how they are non-superimposable mirror images. Then the capital L, capital D forms, so which are more related to the optical isomerism that exists. Then the concept of epimers, how they are different from enantiomers or other type of isomers, anomers, and how they are different. So make a chart if that's what helps you. Look at one example of each and go through these lecture notes again, or make your own notes and make sure these differences are clear, because they sound very similar. You might remember that they are just about carbohydrates, but the subtle differences between them, they are important. So go through them a little bit carefully. Then the concept of muta-rotation, and like I said about disaccharides and the different types of linkages that are present, whether it is an alpha glycosidic linkage, beta glycosidic linkage. See what is the type of linkage that is present in a nucleotide triphosphate NTP. So that's why I said go through those bonds again, because you might be asked about it. Then some polysaccharides, how they are formed, how from one monomer you have a disaccharide or you have a polysaccharide, how is that linkage being produced, and what is the way in which you can actually study them. Like this, you can use Benedict's test, what is the reagent that is used, and how you can actually trace it as well. So all of this can actually be performed. Then in the third and fourth part, what was taken up was cellular metabolism. In depth, I'll not go over this. First, we looked over the process of glycolysis in quite a lot of depth. Then okay, I'll not go through the steps. First, we went through glycolysis. Then we went through the TCA cycle, which is the the Krebs cycle as well. And then eventually energy is being produced. Then we came over lipids. So we talked about nucleic acid, we talked about carbohydrates, then we talk about lipids here, how glycerol is the basic molecule. From there, you get fatty acids. They can be saturated, unsaturated, branched, or substituted, how their structures are different. The number of carbons which are present would differ or would decide the nomenclature of that fatty acid. So again, another table, slightly less clear table, but yeah, still very, very important. So go over that as well. Unsaturated fatty acids can have unsaturation in different positions as well. There are different classifications. How triglycerides are the most abundant. We talked about that. What are their functions? How are they different from waxes, etc.? And go through the different classifications, phospholipids, sphingophospholipids, each of their different functions. If you remember, we had some questions from here as well in the assignment. So make sure you go through them, like how dipalmitoyl lecithin it acts as a surfactant. I think that was the question which was present. So make sure you go through that. And there are glycolipids, there are different functions, glycosphingolipids, how those are different, sulfolipids and how those are different, and some diseases. Again, another table, lipid storage diseases, examples. There are only 3 examples. It is relatively easy to remember. But oh yeah, there were more here. I think a few of them are common. Yeah, try to remember as much as you can. I think not many would be familiar to you, but yeah, do your best. Try to remember as many as are possible for you. And then we have cholesterol and its job, how LDL and HDL are different. Which of them is the good cholesterol, which of them is the bad cholesterol, where they are present, what is their function, why are they present. So all of that, go over all of that. So yeah, then in week 5, I think we can do till week 8 or 9, maybe. The following are relatively recent, but yeah, let's see. Okay, in week 5, we took up proteins, individual amino acids, their structure. And if you remember, I've already told you some structures that you should particularly try to remember. Look for questions like look, when you're reading through the text, also try to look at them as possible questions. It enriches your ability to understand or to remember some details. Like, for example, which was the first amino acid that was discovered? It's easier to remember, right? If you just stress over questions like this. There are 1-letter code, there are 3-letter codes. Remember those, the different classifications. I think you're already quite familiar with how they can be studied, the technique of chromatography, then how you can analyze them, where they would stop migrating depending upon their molecular weight, depending upon the functional group, the stationary phase, etc. So go over that. Then these are the different steps that are taken during TLC. And TLC was also there in the question as well. Then the process of detecting it. Then proteins, how the Ramachandran plot is formed, what are the dihedral angles, which are phi and psi, where the presence of an amino acid in a particular position, what does it indicate? Which is the only amino acid which can be present anywhere in the Ramachandran plot, that is glycine. So questions like that, you look at them, like you look at what you're studying in that way. Then we moved on further into the protein structure, the different levels of protein structure, primary, secondary, tertiary, and quaternary. How you go through each of these levels, how a single linear sequence, it can form all of these different uh, configurations or conformations to form the ultimate functional protein as we know it. Then how is a protein sequence studied? You go through the process of breaking the disulfide bonds, then cleaving the polypeptide chain into little fragments. Then you sequence each of these, and the process of sequencing, what are the reagents that are used, what are the techniques that are required for the process of sequencing, the process of Edmond degradation as well. So what is the concept behind it, how it has a limitation on the number of amino acids that can be sequenced in this, what is the purpose of sequencing, go through all of that. Then the second level structure, secondary motifs which are possibly formed, alpha helix, not motifs, the secondary structures which are formed, the alpha helix and the beta sheet. From there, how the tertiary and quaternary structures are formed, and the experimental methods like X-ray crystallography, NMR spectroscopy, which can be actually used to determine the 3D structure of any target protein. Those can also be studied. Then the the methods also that can be used after in silico techniques have been developed. They include homology modeling as well. You can blast a particular sequence, you can look for a template, and based on a template, you can predict the structure. And now, in fact, there has been further progress, and we have an AI-based machine learning algorithm, which is called as AlphaFold, that actually does it and does a pretty splendid job at it also. And yeah, it is evolving, and we might have more and more softwares in the future. So that was this discussion. Stay. This is not good. Yeah. So yeah, then came the different types of functions. So proteins, if you just think about it, can do almost anything in a cell. Every single enzyme, every single receptor, or the majority of the enzymes, majority of the receptors, they are all proteins. So their function ranges from everything to everything. So I don't think I'll be going much into detail about this, but yeah, here you have enzymes, the different types of functions, how gaseous exchange is carried out, the etc. These that entire week actually looked at topics where we understood how proteins can form different functions, how they can undergo sorting and vesicular trafficking, and yeah, that was looked at in that entire week and its lectures. Loading really slowly. Yeah. Then we came to the central dogma of life, DNA to RNA, RNA to protein. Then some anomalies to the central dogma were also discussed. Then we understood each of the processes: replication, transcription, and translation. The anomalies are reverse transcription. Yeah, again, another table you should look at a little carefully. Some normal circumstances, some special additional possibilities that also exist, but some which cannot. I think it was also an assignment question. So look into that. Then RNA replication, DNA to protein directly in a in vitro cell-free system is possible, but of course it's not possible in vivo. So those are trick questions, do not get them wrong. Then we looked at the process of replication, each of these bases, the complementarity of DNA, what are the different components that you require. You need a template, you need an enzyme, you need entities, and altogether you can synthesize DNA. So this is the replication machinery. What are the enzymes which are also acting in addition to the polymerase, like the gyrase, which unwinds the ligase, which joins additional fragments, single-stranded DNA binding protein, where it is present, how does initiation occur, how does elongation and termination occur, what are proofreading steps that are operating. So all of that was part of the replication process. I think after that, we move towards transcription, if I'm not wrong. Okay, we move towards PCR, again, Carey Mullis. So I think this was a very detailed lecture. I'll not go into it, but yeah. Okay, internet is unstable. Okay, yeah, I got disconnected. Yeah. Okay, so something is the issue with my network, but yeah, that's what we were discussing. I think I'll wrap up a little quickly. But so instead of going through those topics, I think what we can look at are just the overall topics here. So we did central dogma, we did replication, we understood polymerase chain reaction, we understood the process of transcription and translation as well. So what happened was okay, so after a cell has undergone replication and it has doubled up all of its genetic material, all of its DNA, what it will now do is it will convert that DNA into mRNA, messenger RNA, so that it can be translated into protein, which will be able to execute the necessary functions inside the cell. So that is the second step. Then coming after that is the process of translation, which is where the ribosome comes in. So what you need to make sure you understand is that the deviations from it as well. So you are not just getting not all of the information that is inside DNA, it's getting converted into mRNA. There are other regions within DNA which are non-coding, which do not encode for proteins, but they might encode for something else. They might encode for an mRNA which has a regulatory function. And some examples of this are LNC RNAs, which are long non-coding RNAs. There are tRNAs, rRNAs, which are transfer or ribosomal RNAs, respectively. And therefore, every material or every information inside the DNA is not just to make proteins, but it is also to regulate the formation of those proteins, to control other processes, to act as checkpoints. And they have a number of different functions. So at every step, there is some deviation from the central dogma, and understanding some examples of those would actually be beneficial for you. So these were the different components of the central dogma. After that, in week 7, we had the immune system. What are some of the basic properties, the memory aspect of immune system, the different cells that operate, what are the different levels at which immunity is taken care of inside a cell. Then the process of phagocytosis, how cells are taken up, the process of cell death, apoptosis, the entire vesicular transport, how after a protein is synthesized inside a cell, what happens to it. It goes through a lot of processing. It goes through glycosylation. It goes through sorting. It is packaged inside a vesicle. It carries an address with it, a sort of tag, whether it will go to, whether it will get retained in the ER, whether it will go to the Golgi. If in the Golgi, is it supposed to be secreted out of the cell? Is it supposed to be sent to the lysosome for degradation? Or is it supposed to be sent to the mitochondria? What is its job? Based, all of that is carried out during this process of vesicular trafficking and sorting. And therefore, a lot of control and checking mechanisms are actually put in place in these steps. And yeah, we discussed that during this entire week as well. Following that, in week 8, we looked at the human physiology. We looked at the process of digestion. We went through the entire gut, basically, starting from the mouth, where we understood that so for different components of food, everything that you could consume from there to produce energy at the final step, everything that you eat has to be broken down. And each of the different components, each of the different major parts, macronutrients that we understand, carbohydrates, proteins, fats, they get digested at different steps along the entire gut. For example, like we discussed in the assignment as well, that digestion of carbohydrates, it begins in the mouth by salivary amylase. And over the course of the entire digestive tract, digestion also occurs in different steps. So we understood each of those differences as well. And yeah, so that is what happens. And from there, we also understood the difference in dentures as well. Remember the formula for different types of teeth. Then we went through each of the different linings. We understood the different enzymes that are present. We understood different movements that are present, like, for example, peristalsis, which is an involuntary movement, but it is needed to push the bolus from the mouth to the stomach, and for different sphincters to be present in different places to make sure that once digested food has passed from the esophagus into the mouth, into the stomach, it does not go back. So there is an esophageal sphincter which is present, gastroesophageal sphincter which is present, but when you vomit, that sphincter is not actually working. It's because a lot of acidity has been generated in the stomach, and the easiest way to let it out is to push it right outside your mouth, which is reverse peristalsis. So those are some deviations from the normal situation, of course. And we also learned different equations via which, food is getting digested, how trypsin is working, how chymotrypsin is working, how food is further broken down, how different components are broken down. Then from there, we move towards the small intestine, the large intestine, different parts of the colon, how they have all of that extensive coiling in order to provide more and more, to accommodate more and more surface area in a smaller smaller place. And then final concept of digestion, and from there how energy is being produced further is a whole other concept that was also looked at. And that was the first part of human physiology. In the second part, as concepts of week 9 were looked at. Just zoom in a little bit. Yeah. So from there, we understood the circulatory system, how it is present, how your arteries, your veins, your heart and the lung are all each playing their part in the entire process of circulation. Then, yeah, the basic entire process of double circulation, which is present, and we actually correlated that to our initial classes where we talked about diversity and classification, and how the circulatory system present in their case is different compared to ours, and not just about blood, also in some organisms, which have tube feet, if you remember, allow them to move from one place to another. So that is a different, it's a canal system. It has a different purpose, but yeah, it's still a circulatory system that works in their case. But compare and contrast that to what we talk about here, where we basically talk about blood, how we try to maintain the levels of oxygen, or rather the partial pressure of oxygen and CO2 within the body. And the circulatory system has this immensely important and difficult role to play in this entire process. Then we shifted towards the muscular system, each of the different muscles that are present, how they all have their own job, how different types of muscles execute. When I say types, I mean whether it is a smooth muscle, whether it is a striated muscle, whether it is cardiac muscle, whether it works in a voluntary manner and involuntary manner, how each of them do their job in a different way, and how each of them are required for different purposes as well. So that we talked about. Then we discussed the basic unit for muscle contraction, which is a sarcomere. What are the different components? How that the length of a muscle it contract. What happens during the process of contraction and a relaxation? How muscular system cannot operate just on its own, but a nervous stimulation also has to come in, and that actually controls this process. So we learned all of that during those discussions. Then these topics, we learned about the nervous system, we learned about parts of the brain, how each of them have different functions, how some parts are necessary for memory, some parts are necessary for some other purpose, for balance, for your proper gait, to connect what you're listening to, understand the differences between one X area, and to understand how information from the left side of the brain is carried over to the right, and from right to left. It is the corpus callosum which is mediating these communications, and how you need both of them to be in sync for an individual to be able to survive properly under normal conditions. Then the different how brain activity can be studied, how it's a revolutionary idea, the entire concept of an EEG, and what it is, what each of the different types of waves are representing, how they can be studied, how they can track the condition of a patient who might have, suffered a severe trauma. How if somebody is brain dead, their organ transplant, whether it can be carried out or not. They have really significant real life implications. So understanding those would certainly be beneficial for any and everybody. So that was all covered under nervous system. We also learned, I think, about how reflex actions they work, how a stimulus is generated, what happens at a synapse, how saltatory conduction is carried out because of the presence of a myelin sheath. What happens if a myelin sheath is not present? What are the diseases that are caused? So those were topics that we understood in week 10. And finally, as part of week 11, we looked at homeostasis in a lot of detail. We did it very recently, just 2 days ago. So there we understood how hormones in overall in the entire system have an immense role, where you can fluctuate up and bottom, but your body will always bring you back right to the basal level to maintain that. It might appear stagnant, but it is never stagnant. It is the baseline which is necessary to be maintained for your body to perform its best function in the best time and to be as efficient as possible. So there we looked at the different levels, like we understood how pituitary is at the top, then you have the hypothalamus, and then the pituitary, the anterior and the posterior pituitary. They control different, they release different secretory, they release hormones which act in either secretory or inhibitory manners, and they have their own target organs. We understood the difference between exocrine and endocrine glands, how endocrine glands work, how they're able to signal some information which is far away from the start site via the circulatory system again. And upon reaching the target organ, they can have like even a hormone secreted from the same site can act on different organs and execute different actions, and vice versa. So we understood that. We looked at examples of that, and we understood those things in a lot of detail. And overall, we looked at the concept of homeostasis. We understood that that is the basal state. That is how a cell likes to be, and it will always have mechanisms in place which will try to keep it under those state. And if those checkpoints or those basal maintenance systems are down, if they don't work properly, then what it eventually leads to is some sort of a disorder, which will obviously not be beneficial for the cell, and the cell then tries its best to recover. It has mechanisms to actually recover from a lot of things, which is why all of us don't fall ill all the time, which is why we have most people under most circumstances are healthy. But yes, when the like if you if you think of it as a seesaw, there are some factors which instruct a cell to survive. There are some situations in which a cell is trying to consider, would it be easier to save our resources and undergo apoptosis in order to save more resources for other cells to survive. So when that seesaw is, as long as the seesaw is balanced, it's fine. When that gets tipped in the direction of apoptosis, when there's when too much damage has occurred beyond the cell's ability to actually repair, then the cell opts for apoptosis because that's the easiest option to go for and the more sensible option to go for as well. And that is when you deviate from homeostasis. But under normal circumstances, all of these different systems, they coordinate with each other. They function in a manner which allows the cell to do its job the best and survive the best. So yeah, that is pretty much all that we have covered from all of these different weeks. I think this was a very comprehensive, slightly superficial, but yes, an overall a revision of the different topics. It would not be possible to cover so many different topics today in one session. But yeah, I think that was pretty much the idea I wanted to get across today. For your exams, I have 2 suggestions. I think I gave some of it already, but yeah, be patient when you answer your questions. Don't rush into anything. Don't try to finish as quickly as possible. You will have more than enough time to answer your questions. I can guarantee you that. But be careful. Read your question properly. There will always be some which are trick questions, which will, among a sea of information, it will try to ask you a very simple thing. So try to eliminate all that unnecessary information and focus on what the question is really asking you and then attempt it. As much as possible, try the process of elimination, which I think I discussed in the first session as well. Try to eliminate as many wrong options as you can. If you know the answer to question, very good. Don't even have some confusion. Always try to eliminate incorrect options as far as possible and narrow down to the correct one. Do your best, attempt calmly, and do very well. I wish you all the best, and thank you for attending all of the sessions. And for being patient. These were quite long. So this will be the last session. And my internet connection is also really unstable today. But yeah, I think that would be, oh, you're most welcome. That would be it for today. So all the best for your exams. And yeah, I think I'm going to end the meeting now. So